It's really nice to have you all in one place again. It's been a little while since we've had a big general update, and we have some really exciting data and updates to give you today. So I'll go ahead and get started. So today we'll be focusing on giving you some short updates on the work stream. We're going to talk about Eurotalks and also gather some interest in a specs library. But the majority of the meeting will be passed over to our postdoc, Jess Ewald, who will present the data review on some of the Axiom data. So really exciting to see this work that's come out of this collaboration with OASIS and Axiom, and we'll let her talk more about that as we get there. And then I'll just close with some upcoming schedule for 2025. So first, I wanted to kick off the meeting by just talking about a upcoming meeting, and that's Eurotalks. And so, as many of you know, we have a global presence with OASIS, and we're excited to attend a Eurotalks so that we can engage with our European and global collaborators. So in 2025, which is located in Athens, Greece, both David Roquier and I will be presenting a session called Fundamentals and Evolving Uses of Cell Painting and Toxicology Studies. And we have a really great lineup of speakers, so we're excited about this. We're going to cover cell painting through a breadth of industries and also focuses. And as you'll see, both David and Jess, these are all, I think everyone on here are members of OASIS, but Jess and David will be focusing on OASIS, and others will be kind of bringing their perspectives as well. So next I want to pass it over to Bujarki. So we have the opportunity to give some influence into the program for Eurotalks for 2027. This is because it's located in Sweden, where AstraZeneca is also located. So Bjarki, if you're on, I can let you speak to this. Okay.
Thank you. Yeah, I just wanted to take 60 seconds out of everyone's time. So I've loosely involved in a couple of sort of committees internal that are discussing potential topics here. Multiomics, cell painting, and AI is something that we're very interested in, but it's not like AstraZeneca is dictating any agendas here. But I thought maybe we could come together and come up with a couple of topics that we would like to focus on, and maybe sort of organize a track together. It's sort of 2 years out. Or 1.5 years. So we have we have time, and we also have sort of in 2 years we would hope to be a little bit further along than we are right now. So I was thinking about topics in the context of sort of pie in the sky, replacement of animal studies with AI, multiomics, and advanced stem cell model or cell models. Sorry. And here I was thinking maybe it would be an interesting sort of juxtapose both regulators, industry, and academia in this context, and map out where we stand, what is specifically missing, and how can we remove those blockers that are potentially in the way. The other idea that I had, I think now looking at the schedule for 2025, it's already being covered, and we're all thinking in that space. I wanted to ask what people think about this, and if there are additional ideas about what we might want to talk about in 2 years' time at Eurotalks.
Thanks, Bjarki. If anyone wants to unmute, you're welcome to talk, or you can type in the chat as well if you have some feedback right now.
And I left my email there in the slides as well, so if anyone has any ideas, please reach out.
And Bjarki, the idea for this, it's not just individual sessions. This is more almost like a theme of the meeting, is what you're looking for feedback on?
Exactly. It depends on how, it's a big meeting. So, but I think sustainability and thinking, how do we, I think it's a big question, and I think it's worth looking at it, or critically looking at critically looking at that topic. How do we move away from animal work? And I think a lot of people are interested in this, but specifically in pharma, it seems to be ages away, and I don't think it necessarily needs to be. And I think things like this could push things forward a little bit. So.
Makes sense. We got sounds great from Anne in our chat. I think I'll let everyone kind of marinate on this, and I can send it. Oh, wonderful. Terry.
Yeah, no, just a really basic one. Was just wondering, in September 2027, the OASIS consortium will be over. So is it part of the sustainability of the project itself, or how do you see that?
No, a great question. I was thinking rather we have sort of a group of like-minded people here. So I thought, there would be an opportunity for us to come together, even if this is, even if it might be officially over at that point. Yeah.
And I'll just make a note on that although the company, the industry donations are concluded at that point, we may still have work going on. Like we're not just ending when the funds are gone. There should be more data and things to go on. So I hope we're still around in 2027.
Okay, great. Thank you.
Okay, wonderful. Right. Well, if you have any feedback, you can email Bjarki and we'll be in touch. Great. Okay, so for most of you on this call, this is the big slide that I show almost every meeting. I know we have a few new attendees this meeting, and welcome to all of you as well. I'm not going to go through the overview that we have planned out here. These slides will be distributed and you can look at those. But what I like to do every meeting now is give a bit of an update of where we are in our progress to completion. And so I'll just spend a few minutes here. So the text in blue you may have seen before, but really what I want to call your attention to is the text in orange. So, we started over here trying to pull together our public and private compounds. And with that, we spent about a year going through all of these databases, pulling together lists and trying to source these compounds. And we're really at this point now where the Broad has strategically plated they've not only sourced all of the compounds and are finalizing the ones from industry now, but they've strategically plated those compounds. So we are ready to start screening. Jamie will talk more to this in the study design updates, but just wanted to bring your attention there. The assay development for the U2OS and HEPA-RGs has been completed. The primary rat hepatocytes will be upcoming. But with that assay development being completed, we're really excited that the U2OS have begun cell painting screening. So we're generating data at this point. So a big round of applause to the Broad. It's taken a long road to get here, and we're very excited about that. And Jamie, again, will talk to the timeline and when we get to that part of the meeting. Another exciting update is that we had a collaboration with Axiom, and they screened OASIS compounds through human hepatocytes. And with that data has been completed and analyzed mainly under the direction of Jess Ewald, and she will be presenting that data today. And yeah, I wrote that here again. And finally, we have two publicly facing publications. One will be Jess's and Axioms that will be coming out. And then Andreas and David have led the preparation of a strategy paper, and we're hoping to get that out in the next few months as well. And for those of you who are interested, the compound lists that I mentioned are all available for the public, and they're listed on our website. Okay, so with that overview, we'll go ahead and get into workstream updates. And so if Jamie wants to give an update here, let's see. Jamie, are you on?
I'm here, but this is compound selection and data extraction. So I think this is not me.
I'm sorry.
Yes.
I'm sorry. Okay. Real quick, I can just give a quick overview. I really gave you the big picture here, but we have established this comprehensive list of compounds, and those are all procured and at the Broad. I said this, so I'm just going to show you. This is from our website here. We have both the full compound list that is available for download, and then also the prioritized subset. And so just to make you all aware, for any partners that are not able to run the full, 1,400 OASIS compounds, we have a prioritized subset, and these compounds were selected through a variety of parameters, everything from how expensive they are, how difficult they are to procure, to the more important side of having some in vivo data that we can benchmark to. And so this is a really nice subset. It looks like, yeah, there's about 500 compounds here that we're asking if people want to run a smaller set that they all run the same compounds. Okay, sorry, Jamie, I'll jump to you here.
All right, great, thanks. As Chrissy said, it took us a long time to get to starting, but we have actually started screening itself. So thanks to the compound selection team, we were able to get 1,400 commercially available compounds, both from enamine and from Molport in. All 1,400 compounds will be screened at 8 doses in 3-fold dilution, depending on the solubility at which we could get the compounds at top solubility. So we aim to get almost all the compounds at 100 millimolar in DMSO. Anything that failed at 100 millimolar, we went down to 33.3 millimolar, and if it failed there, we went to 11.1 millimolar and stopped there. The majority of the compounds went into solution. I think there were a handful that didn't fully go into solution at 11.1, so we just kind of took the supernatant and ran with it. There, Chrissy's list that is publicly available, we will attach all that solubility info and what we were able to get to soluble at and what we will be able to screen it at. We'll attach that to those lists down the line. So we have done both U2OS and HEPA-RG assay development. We had used some, for the HEPA-RGs, we had used some cell painting controls, compounds that are known to elicit a cell painting phenotype, and then non-toxic controls. We have also set up how to do randomization of the compound location on the downstream assay plates, so that no compound or dose is ever in the same well position across the 4 replicates during the screen. And then thanks to Srijit and Jess, we actually took the 1,400 compounds, and when we plated them in our source plates, they prioritized and arranged the best way to lay out those compounds so that we didn't either end up with too many toxic compounds, too non-toxic compounds, and also that prioritized list is on a specific set of source plates so that downstream, if we need to run just that subset, we know where those are. So as Chrissy said, during our pilot testing of the assay development of the HEPA-RGs, we sent the supernatant to Nomics Bio to test the proteomics arm of this to see how that looks. That data actually just came back on Friday, so we'll be setting up a meeting to see how that looks so that we know that informs us how we handle the proteomic side during the screening itself, and we'll be doing assay dev with BioSpider on the transcriptomic side to make sure that arm works as well. Chrissy, you can go to the next slide, I think. Yeah, so as you said, it took a long time to get the commercial compounds in. Those are running, and as of last week, we finished the last batch of U2OS cell painting, and now they're all on our imager collecting data. The way the screen is designed is that right now the 1,400 compounds are across 35 source plates in 8.3-fold dilution. The top dose is 300 micromolar for as many compounds we could get to that top dose. If not, it's 100 micromolar or 33.3 micromolar. Again, that depends on what the top solubility of that each specific compound is. Each compound plate was run in singlet in a batch. So we ran all 35 compound plates in batch 1 and then had a new batch of U2OSs, and that was batch 2. So we have 4 singlet runs of the U2OSs to make up the 4 replicates, and each of those batches have a randomization of the compounds across the plate. So that was all done last week. One massive hat to my research scientist, Adam, for just crunching through what was probably 1 billion cells to get through all of that screening. And like I said, that is all in image acquisition right now, and rolling up to Ann Carpenter's and group for data analysis. And whoever said winter is coming, yeah, that's what I've been telling my poor group for 6 months now, is that this winter is going to hit hard, and here it is. We're also pulling in the industry donated compounds at this point. We have a handful from Bayer, GSK, and Cortevas is coming in this week. I think there's more coming in from Bayer, and then Merck, I think, is also donating some as well. So we're trying to get all those in so that our compound management can handle that, get those on source plates, and then we'll run those through the U2OSs. And we worked with Lonza to get bespoke media made for us. So we have one batch of media for all the HEPA RG cells. We have one lot of HEPA RG cells sitting in our freezer right now. So once all those remaining compounds are in, we will be running through and doing the screening cell painting arm of the HEPA RGs, hopefully starting by March. We'll do the 4 rounds of cell painting to start, and then do the transcriptomics and proteomics behind that. I see Anne's hand just went up.
Yeah, I was just
I lost Anne there for a second. I think that's generally it. And then, sorry, yes. And the last thing is, now that we are putting the U2OSs behind us, the HEPA RGs are coming. We're going to be working with Visicol to get primary rat hepatocytes, and we'll start doing assay development in those. That one's a little bit, I'm going to say, quote unquote, easier, just because they are plating the cells in a ready to use 384 well format for us. So it's just a matter of making sure that our systems work with their pre-plated cells. That one will not run the full deck of compounds just because it's so cost prohibitive. So that prioritized subset that Christine mentioned at the beginning is likely going to be the set that gets run through the primary hepatocytes. And Jeff.
Yes, so now I will not echo, since we're in the same room. My question was about, of course, in a perfect world, we would put the company's contributed compounds in the exact same batches as everything else. That ship has sailed for U2OS, of course, because it took quite a while to get those compounds gathered. Will it be possible for those contributed compounds to be run in parallel for the HEPA RGs onward?
Yes. Yeah, we'll just tag them onto the back end of the run for the HEPA RGs. Yep, that's no problem.
Awesome. Those are certainly more it's more important for those than U2OS, so that's great. Thank you.
Yep, no problem. Jess, did you have a separate question, or was that just Anne hopping on with you?
Nope, that was just we were having mic and mute and everything difficulties.
All right, so I think that's the update from me.
Okay. Well, before I let you go, Jamie, I just want to say a very public thank you for you and your team's hard work. This has been huge, and we're really excited to see the data rolling in. So yes, thanks for all the applause. And I also wanted to open it for questions from anyone in the group. Jamie is our resident person who will answer every single question you have about the study design, from concentration through timeline. So anything that you have, we'll just give it a minute or so.
Yeah. Carrie has a question. Yeah, do you want to go ahead, Jamie? Sorry.
Somebody in the chat said, are the rat primary cells from a single donor? I think they're a pooled donor for the rats, but they told us, and when I had this conversation with them a year ago, that they could keep the same pool for our entire study. So I need to round back with Visicol to discuss with them where we stand on that. And then we'll be pulling Lonza into that conversation as well, because apparently Lonza supports Visicol in making these. So that's my next step. Shantanu.
Hey, Jamie, I had a question about the proteomic data generation. So when we've done that in the past, we've always done it in parallel with cell painting, mostly because of convenience, but also because it allows us to have the data closely paired. I'm guessing we're not doing that this time. I'm just looking at the timeline, the way
logistically, it's easier for us to pair the transcriptomics and the proteomics on the same plate.
Oh, that's what you meant here. Okay.
Yes. So we're going to run 4 batches of HEPA RGs in cell painting alone, and then run another 3 batches that we will split for proteomics. The supernatant goes to proteomics, the cell lysate goes to transcriptomics. So it will all be done one behind another.
Okay, so it is in parallel with one other assay, which works well. Okay, great, thanks.
And just logistically, it's easier for us to do the transcriptomics and the proteomics on the same plate, rather than try to pull the proteomics off the cell painting plates as well.
Interesting. Good to know.
Yeah.
Yep. Thanks.
Yeah, I see another question from Bullock. Are you confident in dosing and exposures will be sufficient to trigger the mechanism of action that underlies the the liver toxicity for most compounds? I can maybe start and then turn to Jamie and the steering team. For this, a lot of what we did was sort of based on choosing a high concentration and then the solubility of it. So we have a kind of high end, and then we're doing 8 half log dilution. So we cover a really large space in the concentrations. So we would think for most of them, we would hit it, but given it's a large screen, there might be some that we do not. I think that is where some follow-up and, the IVE might come in of, did it not get into the cells? Did it absorb to plastic? Were we not high enough? What's the human equivalence? So I think there will be, the subset that we don't see what we expected that we need to follow up on. But that's kind of where I start. Maybe turn to Jamie and then anyone else on the steering team that wants to attack that.
Yeah, we relied really on what this committee told us in terms of dosing. I know some people wanted to go higher on the dosing, but that's just not logistically feasible on the compounds themselves. And we are exposing for 24 hours, which again is more of an acute toxicity. In our assay dev in the HEPA RGs, we definitely saw some effect of some of the compounds we chose. So I'm hoping, but I don't know truly the answer to that.
No, thank you. I was just curious because actually in JUMP, I believe we did 48 hours, not 24, which is actually a double, right? And of course, I would expect, for instance, a metabolically active cell to be more sensitive than a U2OS cell, right? So, yeah, more of a brainstorming question than anything. It's smart to say it. Just yeah. Thank you.
Per Jess's request, we did put a JUMP plate compounds on there, so there's about 310 of the JUMP JUMP target 2 plate. So that in itself is on there for both the U2Ss and will be there for the HEPA RG. So that's an anchoring point, I guess, against previous data. But this is kind of a try it and see experiment.
Thanks, Ross. Thank you.
Thanks for that. Any other questions? Okay, we'll go ahead and move on. Thanks, Jamie. And now we'll get a quick update on QIVIVE from Jess.
Hi, everyone. Yeah, for the QIVIVE, we were working on this for a while before the data arrived, when the postdocs had more free time. So we outlined the overall strategy. Basically, just what is the most complex set of models or the most sophisticated approach that we could feasibly do for all 1,500 of our compounds, sort of regardless of their prior information. So we chose a set of open source models that we can use to both QSARs for estimating pharmacokinetic parameters, and then also a basic 3-compartment pharmacokinetic model for trying to estimate the administered equivalent dose that an organism would have to be exposed to in order to reach the concentration where we see activity in our in vitro screens. So we've set out an overall strategy for doing that and identified the tools, and we've talked with John Arnott with his Easy Suite tools for some sort of complementary approaches for estimating some of these parameters and doing this modeling. And yeah, we'll use these models once we have all of our data in and we want to do these final comparisons to the in vivo data. Yeah, this is the 6 kind of main steps for what the strategy is. The first will be to identify the concentration at which we see activity or the activity that we're interested in using each of the cell models and data types. Next, we'll be applying in vitro mass balance modeling to try and correct the dosed concentration or the nominal concentration for solubility and partitioning factors, so we can try and understand what concentration in the actual cell caused that activity. Next, we'll be estimating 2 main parameters using the OPERA QSAR model, the fraction unbound in plasma and the internal clearance. And then we'll be estimating both the human and rat administered equivalent doses using the HTTK-R package. Finally, we'll be kind of just taking all the in vivo points of departure or the concentration where we see effects in vivo from the publicly available rat data and then comparing this across the cell-based measurements and the publicly available in vivo data to see if we're generally, detecting mode of action signals and general bioactivity around the same concentration, and therefore it could be helpful for setting a safe concentration for exposure.
Thanks, Jess. Okay, and finally, the data management and integrative data analysis workstream has been meeting. They met a little bit in the fall just to kind of start kicking things off. And so really what we've done is we've outlined the data management and analysis pipeline, but this team will really come into action soon as the data from the U2OS cells are coming in. And the other part of this is that the Axiom data had been procured, and Jess will be presenting that analysis today. So are there any questions about any of the work streams or anything you've heard so far? Okay, wonderful. So before we get—
There's a question in the
there's
chat.
one in the chat. Oh, thank you for that. Okay, so I see Josh. Oh, I see John. Is there general awareness to the potential confounding effects of DMSO? And Jamie said the DMSO load is 0.3%.
Yeah, sorry,
John.
I was just explaining to John.
Yeah,
so,
it's okay.
By design, and part of the reason why we couldn't really go higher is that we didn't want to increase the DMSO load too much to the point where DMSO itself is toxic. So right now, the entire assay is designed so that the load is 0.3% DMSO. And I think in discussions with Josh and Jess, who have done HEPA-RG cell painting, that's pretty tolerable and should not have effects on hepatocytes in and of itself. And Josh and Jess can weigh in if I'm incorrect there. But we tried to keep the DMSO load low to minimize any vehicle effects on the assay itself.
All right, great. Okay. So before we jump into the data analysis, I did want to give Johan an opportunity to talk a specs library. And so just to tee off, we've chatted before about how challenging it was to source all of these compounds from one place. It wasn't possible. And so what we've talked about is potentially putting together a library that would be available for purchase for any partners who want to run the OASIS compounds. So I'll go ahead and let Johan take over.
Thank you very much. Hello, everyone. Yeah, very brief introduction. I'm new to the group, so my name is Johan Thijhuis. I've been working for Specs for 30 years now, and I'm an organic chemist by training. So Specs, I'm not sure if you're familiar with the company, but Specs has been in compound libraries all its life, so like 40 years almost. Initially as a compound broker, but the last 2 decades being a provider of compound management services. So we deal with compounds and all the tedious jobs that you heard of procuring this library. We have done that for our lives almost, so we're quite experienced in that. So we manage now compound collections from pharma companies and other organizations. So we do everything that's included in the whole logistics process from the moment a compound has been synthesized up to the screening, the assay ready plate. So it involves storage and reformatting. So a lot of weighing and liquid handling is done. It involves also procurement of compounds from many different sources. So this ended up, we got a question in 2019 from a number of organizations to make the broad repurposing library available. So we procured those compounds on something like 5,500 complex molecules, all known compounds with biological activity. So we did that for a number of people. And then later on in 2021, we also procured the cell painting sets, the MOA and Target that we just mentioned. So we made that available as well. So in line of that, I got a chance to talk to Ann Carpenter at a meeting, and the idea came up to make the OASIS compound library available as well. So that's the reason why we are at the table now. So maybe the next slide. Yeah. So our idea was to make this compound library available. So the 1,400 compounds, more or less. So it will depend on how available these compounds are, if they are controlled substances or not, if the quality is okay. So the number is not fixed yet, but that's the idea, to make that whole set available. We use various sources for that. So we have our own network of suppliers, but also as part of an e-molecules group, we also make use of that network. And typically we can find sometimes better options for compounds in better quality, better prices. So that's one of the tasks that we will then do. We want to make sure that all the compounds in the set also meet the QC requirements. Typically, we take 90% plus. This can be discussed if it should be higher or not. We have an in-house NMR and LCMS to do all the analyses. And then we make sure that everything is in the right format. And what I learned now, there's also a layout of the compounds in the plate, so we can also follow that. The concentrations also starting with the 300 millimolar down to 100 or whatever is possible. So we will then follow the same steps as Jamie just explained to get the highest possible concentrations of the compounds in the plates. Typically, we are used to dealing with hundreds of different organizations and all having their own in-house preferred formats, so we can easily adapt to that. So it can be 96 or 384 well plates for whatever plate type people have in mind. The volumes can also differ. So from 100 microliters up to a milliliter if needed. So depending on the individual requirements that people may have, we can easily accommodate that. And we can also have backup materials available for resupplies or for backup or QC questions. If there might be something questionable in the plates, then we can also trace that back to the original material. So this is a very quick and brief introduction. So we would like to do this. If people, you can all raise hands here, but I think it's easier if you can send an email maybe to oasis@specs.net if you have questions or a specific format that you would like to inquire about, and then we can provide the details whether that's possible or not. But generally, we have been able to always provide the formats and the compounds that people are looking for. So this would be a very nice opportunity.
Thank you.
So,
are there any questions right now? And yes, will we add this to the OASIS project website? We can definitely do that. I think that would be really great for our partners.
One thing Jess had raised at the last steering team meeting where we kind of previewed this was that there's a pretty big overlap with this library and the first ToxCast library. Is that right, Jess? So that might be one thing to note, because that's not something you can just go purchase, like the EPA ToxCast library. Jess, am I correct on that?
Yeah, I have some more details. I don't actually know which version of the ToxCast library it overlaps with, but we have very high overlap.
So that might just be another, I don't know, quote unquote selling point, that that's not something people can go purchase, but this would be a way to overlap. But maybe Josh knows more. Oh, I can't hear you at least. Can others?
No.
Sorry, how about now?
Yes.
Yep.
Oh, sorry about that, microphone problems. Just kind of a point of caution on that point. A lot of folks refer to the ToxCast chemical library as if it's a static thing. It's not a static thing. It's constantly evolving, shrinking, growing. It's no longer widely available, but I think it may be a better way to phrase it differently. Like this compound library we're talking about here has chemicals that have been tested as part of the ToxCast program, and instead of saying those that are present in the ToxCast chemical library. And we don't use version numbers.
Yeah, that's a really great point. Thanks, Josh. Any other questions? Okay. Well, with that, I'm going to pass over the screen to Jess so we can hear about some exciting data generation or data analysis. Jess, you can take over now.
Okay, can you see the presentation?
Yes.
Yeah, okay. Cool. So yeah, we're very excited that I just need to figure out, that the first OASIS cell painting results are in. So this was screened by the startup Axiom, and I put their website there, so you could go check them out if you want to. They took the subset of our OASIS compound list that was available at through MedChem Express. So that was 1,085 compounds, and they screened those in primary human hepatocytes that were pooled from 5 donors. It's a standard cell painting experimental design, and they did 8 concentrations with 2 replicates each. And then they also measured the LDH and MT cytotoxicity assays for each well, so that we have that nice kind of parallel data. With this set of data, we are mainly for this analysis focused on cytotoxicity and mode of action analysis. Of course, like the overall goal of OASIS is to benchmark all of this with in vivo toxicity data, but sort of in an effort to not scoop the whole big project paper, but still wanting to analyze this data sooner rather than later, we sort of focused in on cytotoxicity and mode of action. So the main questions that I'll address in the results here, first trying to understand what concentration do we see morphological phenotypes, where we are kind of looking at general bioactivity output endpoint, where we're summarizing each cell painting profile as a Mahalanobus distance from each exposure to the DMSO centroids. And then when we do concentration response analysis of this sort of summary, we can get an overall picture of the concentration where we see any general morphological impact. And then we're interested in the sensitivity of that. So comparing it to sort of general cell stress and cytotoxicity that we can assess with these other assays. We want to know beyond cytotoxicity what different biological signals we can detect. So we can attempt this with sort of unsupervised clustering, including compounds with known mode of action. And this is the approach that's mainly been taken when using cell painting data to screen large compound sets. But this has some problems, especially, we're looking at toxic compounds, so that impacts cell count. And so we often will see that the overall clustering of compound profiles might be driven by this general cell stress or cytotoxicity, even though we know other more specific and interesting stuff is going on there. So supervised analysis is specifically supervised machine learning is a really good way of tackling this problem. So we're trying to apply some of these methods to this data set. And then finally, since this is the first batch of data that's coming in, we wanted to look at some more technical questions around what is the best way to process images for mode of action prediction. So including looking at different ways of handling multiple concentrations, and then also looking at different ways of turning images of cells into numerical profilers, or numerical profiles, including Cell Profiler, which is this traditional software that our lab here has developed, and then also newer deep learning methods for coming up with numerical profiles that describe the morphology of cells. All right, so the first set of results show that high throughput imaging detects compound bioactivity at lower concentrations than cytotoxicity assays, as we would hope. So when we're talking about assay sensitivity, I think we can talk about that in 2 different ways. We can think about the number of compounds that are active in an assay, and then also the concentration at which the assay detects some sort of activity. So according to both understanding of assay sensitivity, we see a clear ordering where cell painting is the most sensitive, followed by the MT assay, followed by cell count, and then followed by LDH. And this makes sense when we think about assay biology. So the LDH assay is assessing membrane damage. So it's really assessing this kind of subset of catastrophic cell death when cells are just sort of exploding or bursting. Cell count is looking at the balance of any cell proliferation or cell death activities. So we would expect to detect more activity there than for LDH, which is more of that subset of cell death. And then the MT assay is looking at mitochondrial activity. So it's really looking at the flow of electrons through the electron transport chain. And so when we see higher oxidative phosphorylation, we get higher MT readouts. And so we usually see decreases in that as a result of cytotoxicity. So it's used as a cytotoxicity assay, but you can also just see other impacts on metabolism there. So it makes sense that that, again, would be more sensitive. And then finally, cell painting captures a wide range of mode of action. So we would expect this to be the most sensitive. Do we want to do questions throughout the presentation or at the end? I'm not sure what's the best.
whatever your preference is, Jess.
I can answer questions with the understanding that we might, I'll stop answering questions if it starts taking too long. So yeah, go ahead.
I was just wondering, are these points IC50 values, or what are the activity or the concentration that you're looking at?
Good question. It's analogous to the IC50, but it's called the point of departure. So instead of looking at exactly the 50% activity, we're looking at sort of the earliest point where we detect activity. So
wherever you see the earliest point of activity.
Yeah.
Okay.
I had slides.
Is there a threshold for saying 10% activity and that's active, or it's like?
We're looking at like the 95th percentile of DMSO. So for each concentration response, we have like 60 DMSO that are that we can compare to. So it's pretty robust. Yeah. Okay, I will continue. All right. So again, we see clear morphology signals at a lower concentration and for a higher number of compounds in these other assays. I did want to call out some interesting kind of corner cases here. So I said in the MT assay, we're mainly looking at decreases associated with cytotoxicity, but for a small number of compounds, we saw a robust and significant increase in the MT readout. So I dug into, I found the 4 that had the strongest trend here. They're shown here, and 3 of these are pharmaceuticals with off-target impacts in the liver. That have been mechanistically linked to oxidative phosphorylation decoupling. So when this happens, the ATP production is far less efficient, and so oxidative phosphorylation rate ramps up to try and compensate, and so that could explain why we see an increase in the MT assay readout here. This 4th one is really interesting. It's the most potent and has the highest maximal response, but it's not a pharmaceutical that was under development or whatnot. It is an industrial compound that is produced between 50 to 500 tons per year. I looked it up on the ToxCast dashboard, the CompTox dashboard, and there's over 100 publications in material sciences journals, but not really any investigation into the toxicology of this compound. It was included in several high throughput tox screens, including ToxCast. And from these results, we see that it has activity in 12 different assays that are assessing binding to 8 different nuclear hormone receptors. And this is occurring at concentrations that are far below cytotoxicity. So we think it's really interesting that, this prior source of screening data found that it could have maybe some impacts in the cell through more specific mechanisms at concentrations far below cytotoxicity. And we see the same thing here. So we're interested in kind of looking into this more. Okay, so next, I'm going to go a little bit more into the supervised analysis, where we're first looking at these paired cytotoxicity assays, so this MT and LDH exposures. And now these are matched at the well level, so for every cell painting profile, there's a corresponding MT and LDH assay that we can analyze. So first of all, we wanted to just sort of look at the ability to predict the MT and the LDH readouts based on the cell painting profiles. But we know that cell count and well position are tightly coupled to these assay readouts. So what we really wanted to know was whether, and we know that cell painting profiles encode both cell count and things related to the well position viability of the cells. And so what we really wanted to know is it capturing more than these 2 things? What additional signals could it be capturing? So to do this, we predicted the MT and LDH assay readouts based on cell painting with an XGBoost regressor model. And then we compared that to 2 kind of baseline measurements. One is retraining that same XGBoost regressor model, but using only well position and various cell count metrics to see how much we could predict based on just that. And then we also did a simple comparison where we have two replicates. So we took our predicted value as just the value of the replicate, kind of get an R squared between our replicates. And what we see here is that cell painting does not in any way outperform cell count and well position for LDH. But this measurement was extremely impacted by well position effects. And so you see even the replicate does not have very high correlation at all. So this might not be surprising here because we know that cell count is super tied to LDH and there was a pretty high level of variability in these data. For the MT readouts, the consistency across replicates was much, much higher. And here we do see a significant increase in our ability to predict those readouts based on cell painting compared to just based on cell counts and well-positioned information. So next we wanted to know like what additionally is cell painting capturing. And so for this, we used something that we're calling target enrichment analysis. So this is where we want to like look for patterns in groups of samples. So what this is it's sort of like gene set enrichment analysis, but instead of doing it on sets of features, we're doing it on sets of samples. So what you can think about it is that like for all of these compounds, we have annotations in the literature that say this compound hits this target or this compound is associated with activity in this target. And so we curated all of these and then we can look at a group of samples and say compared to all of the samples, are there more compounds targeting a specific gene than in the overall background? And so we can sort of calculate a p-value for how enriched a group of samples is for compounds that hit a particular target. This is just kind of a very high level way of trying to pick out patterns in these groups of samples. And so what we did is we compared the error of our prediction based on cell painting to the error of our prediction based on cell count and well position. And so that allows us to find a group of samples that are better predicted by cell painting versus those that are better predicted by just cell count and well position. For this one on the left here, we sort of expect that compounds where we're better predicted by cell count and well position to be pretty random and not really have any pattern. And we do see no enriched targets here. So that is kind of increasing our confidence that this analysis makes sense. For the ones that are better predicted by cell painting than by these technical factors, we found that these were enriched in 480 different targets. And these targets were mainly in pathways related to the cell cycle and various forms of signaling. This is an extremely high level summary at the top of this slide, just because I don't really have time to go into too much more, but there's a lot of information here that I think we could dig in to understand exactly what morphology is capturing that something simple like cell counts aren't. All right, so next, we wanted to go beyond just cytotoxicity outcomes and look at the ability of these morphological profiles to predict activity in external assays. So for that, we turned to the ToxCast results. So as Chrissy alluded to, there's a really good overlap between this compound list and ToxCast results. So for 963 of these compounds, they were tested in some way by some ToxCast assay. ToxCast is huge. They're assessing thousands of endpoints. So we went through all of those to try pick out groups of assay endpoints that were the most relevant to our experimental design or what we were interested in. So we curated a subset of assays. First, for many of the assays that ToxCast ran, they ran cytotoxicity assays in parallel. And often the cytotoxicity assays were run in different cell types and cell types from different tissues. And often they were repeated many times for the same cell model. So instead of having like an individual cytotoxicity assay that's repeated so many times for the same cell type, we sort of aggregated these at different levels. So we aggregated the cytotox readout for each compound at the cell level, and then also for all this related cell types that are derived from the same tissue. So there's kind of 48 different calls there. So we wanted to see how well we could predict cytotoxicity, even if it was measured in a different cell type or tissue. We curated 292 cell-based ToxCast assays, and that included trying to address the cytotoxicity confounding, where if the cytotoxicity threshold was similar to the activity call AC50, we did not consider that a hit for a specific assay. And then finally, we also curated results for 72 different cell-free assays. So these are ones that are assessing binding or enzymatic activity for just a protein. We wanted to be able to try and predict the activity in the ToxCast assay. Each of the ToxCast assays is a concentration response format, and then we also have concentration response format for the cell painting profiles. So we tried to kind of process all of this data to make these comparisons possible. So this is actually an example from our MT assay, but we did the same thing to ToxCast, where if significant activity was detected along the concentration range, we gave it a label of 1. And if there was no activity detected, we gave it a label of 0. So that just gives us 1 label per compound endpoint combination. Next, for the cell painting data, one of the things that we have done in the past when we have a whole bunch of replicates are to create a consensus profile so that you have just 1 profile for a compound. So that's where you would take the median aggregation of all of the feature measures across all of your replicates so that you have like 1 compound to 1 label that you can use for, or 1 compound profile to 1 compound label that you can use for training supervised models. In this case, we had 8 different concentrations, not just replicates. And so I'll go into this later. We did a deeper investigation of this, but as a simple starting point, we said, okay, let's just treat these all as replicates. And so we took 1 consensus profile for all 8 concentrations. And I didn't think that this would work at all because it seemed strange to me to like concatenate across concentrations. But this, gives us a nice 1 profile to 1 label for each compound. And when we do this, it actually worked pretty well, concatenating the information across concentrations. So what I'm showing you here are just the results for when we do this type of processing to our in parallel measured LDH and MT assays, just as kind of a benchmark to see what we get when the experimental designs are perfectly aligned. And you can see first we have a random baseline where the labels are randomly shuffled to just give you a baseline. We hope that this is around 0.5, and it is. So this is just kind of assurance this is working properly. Next, we trained a model on just cell count features, again, as sort of another baseline. And then this is the one that's trained on all the cell painting profiles. So you can see it achieves pretty good performance here. When we look at the ToxCast cytotoxicity assays, we again see this same trend, although everything is quite a bit lower. This makes sense so for example, these ones up here, many of these were cytotox measurements made in liver-related cell lines. So it makes sense that our performance there would be higher. And then some of these are in totally different cell types. So again, we sort of expect this decreased performance. Next, looking at all of the ToxCast cell-based assays, I think this is interesting. The cell count does not have any difference from the random baseline. So we see that for these more specific targeted assay endpoints, the cell count information isn't giving us anything, which kind of makes sense. And we do see a significant increase in performance for the morphology ones where, for many of these, this AUROC is quite low, but it is significantly higher than our random baselines. And some of the endpoints were better able to predict than others. And then finally, for the cell-free assays, we actually see no ability to predict based on morphology or cell count compared to the baseline. All right, so I just wanted to say, this again, we're looking at hundreds of assay endpoints, and I'm in the process of drilling down more to look at specific categories of endpoints to see what we can do better or worse jobs at doing. One of the things that came out is that the androgen receptor binding, there's 9 different assays that assess this, both agonist and antagonist, and we're doing consistently quite well with these, which I think is interesting because in previous studies that have kind of systematically looked at binding of nuclear hormone receptors in cell painting, androgen receptor is not something that we've been able to see. So it could be because we're looking at hepatocytes, but I'm in the process of digging into all of that more. All right, so finally, we're looking at what is the best way to extract information from images for mode of action prediction. So I'm going to go back to that concentration question where I said I thought it was a little bit of a bad idea to incorporate all of the concentrations into one profile. And we thought that filtering out irrelevant concentrations would improve this assay prediction performance by boosting the signals that we care about. So one thing that we could do is only take the consensus profile of all the bioactive compounds. So that's higher than this morphology-derived point of departure. This blue line is where we observe cytotoxicity. So another option would be to take the bioactive but not cytotoxic samples. And the thought was that this one would be best for predicting cytotoxicity assays, and that this one would be the best for predicting targeted assays that are trying to look at more specific endpoints because it wouldn't be confounded by general cell stress. However, when we go into this, nothing beats the all, which was unexpected and not what we thought would happen. And of course, filtering out the cytotoxic samples does decrease performance for these cytotoxicity predictions. And we see this across all here, where we're never doing better than the all concatenation. So I've thought about this, and I think we could kind of explain this in two ways. One is that when we do the filtering, we're removing a lot of the concentrations, and it could be that that kind of decreases our ability to average out the technical well position effects. So maybe we are boosting our signal, but we're losing a big benefit of averaging out these random technical well position effects. Another could be that this Mahalanobis distance-based curve fitting, it's more sensitive than cytotoxicity, but perhaps there's a lot going on at lower concentrations that we're not detecting. And so we might explore more sensitive, perhaps supervised methods for trying to derive a point of departure. Finally, we also wanted to compare deep learning features. So we use Cell Profiler as a default to come up with quantitative morphological features for each single cell using engineered computer vision measurements. These days, there's also all sorts of deep learning models available that we can use to generate morphological images. So we looked at two, Cell Painting CNN, which is a convolutional neural network that was pre-trained on several large cell painting data sets, and then also the Dino Vision Transformer from Meta. And those features are coming at the image level. Interestingly, we don't really see a difference between any of these methods when it comes to the final supervised prediction of the assay endpoints. We see a slight bump for Dino with a better AUROC by 0.02. Of course, this could be significant if that's capturing like one mode of action that the other ones aren't. But overall, we didn't see a huge difference. All right, so finally, conclusions and next steps. First, I think like this data shows that cell painting is more sensitive than multiple cytotoxicity assays and captures many mode of action details. At this point, for supervised analyses, using all of the concentrations is a simple but performant method, and different representations perform similarly. So that will guide our future analysis of other OASIS data sets. For next steps, we're in the process of submitting a preprint kind of showing all of the core analytical pipeline here. And we will submit this to a full journal in very shortly with a few more details added about these specific mode of action explorations that I'm doing. And we will be applying a version of this pipeline to all future batches of OASIS data. Okay, I was trying to get through the last bunch of slides to be on time.
Wonderful job. Thank you, Jess. For sake of time, I'm just going to close real quick. But if anyone has more questions, one, the preprint should be out soon. And two, you're welcome to email Jess. Jess, would you mind throwing your email in the chat?
Sure.
And we can look. There's a lot of questions and comments that we can look through, but I know people have to jump off. So just wanted to let everyone know that we will be kicking off or reconvening our QIV-IVE and data analysis work streams as the data comes in. So I will send out invitations for those when we have those scheduled. And I think Connie put our website in the chat earlier, but this presentation has been recorded and will be uploaded to the website. So you're welcome to take a look at anything we discussed. If you have any other questions, you can reach out to me or Connie. And I think that is 11 o'clock. So I'm going to go ahead and call a meeting, but I really appreciate everyone's input today and presence. And we look forward to a great 2025. So thank you, everyone.